So today we are here for the NPTEL live sessions. Starting my presentation. Have you people joined last time also, or you are fresh? Screen is visible. Am I audible? Good evening, everyone. So myself Sonam Pahuja, and I am here for the week 4 live session of NPTEL course of cell biology, that is cellular organization, division and cell processes. So here we will start with the week 4 syllabus, that is lecture 13, 14, 15 and 16, covering the mitosis part 1, 2, cytokinesis and centrosomes in development, evolution and disease. So starting from the questions. So first question is, which of these scientists coined the term meiosis? Anyone? Anyone knows the answer? Am I audible? Am I audible? Please reply. Hello? Am I audible now? Hello? Now, am I audible?
Yes, ma'am.
Okay. So, myself Sonam Pahuja, and I am here for the live session week 4 of cell biology, cellular organization, division, and processes, which is an NPTEL course. So, starting from the week 4 syllabus, which covers these lectures mitosis part 1, 2, and cytokinesis, and centrosomes in development, evolution, and disease. Okay, now everything is fine. Am I audible and screen is visible, right?
Yes.
Okay,
Yes.
fine. So, starting from the question number 1, which of these scientists coined the term meiosis? Anyone knows the answer? Anyone? So, this is Farmer and Murray. These people coined the term meiosis. Okay. Now, question number 2, chromatids coiling in the meiotic and mitotic division is what? Plectonemic, paranemic, or what it is? Chromatids coiling. Anyone knows the answer? What is plectonemic? So let me tell you, plectonemic coiling is where the two strands of DNA are wrapped around each other in a helix, making it impossible to simply move them apart without breaking the entire structure. Whereas in paranemic coiling, the two strands simply lie alongside one another, making them easier to pull apart. Now, I think it is easier for you to tell whether in meiotic and mitotic division, which is plactonemic and which is paranemic. Anyone? In plectonemic, two strands of DNA are wrapped around each other in a helix, making it impossible to simply move them about apart without breaking the entire structure. तो ये स्ट्रक्चर ऐसे ही रहेंगे प्लेक्टोनिमिक में. और पैरानिमिक में क्या है? पैरानिमिक में two strands simply lie along one side each other and they can be easier pulled apart. Okay, so if these could be easier pulled apart, so which kind of coiling will be in meiotic and which will be in mitotic? Anyone? Please speak up.
Option 4, plectonemic in mitosis and paranemic in meiosis.
In meiosis, paranemic, and plectonemic in mitosis. Sure?
Yes, ma'am.
Okay. Any other? So when these structure are together, it will be in mitosis. And what happens in meiosis? Haploid gametes are formed, so it will be paranemic. Right? So option 4 is the correct answer. Right. Now moving to question number 3. When there is an increase in the condensation of chromatin during the process of cell division, what increases or what decreases? Heterochromatin or euchromatin?
Heterochromatin increases and differentiation of euchromatin. I think only heterochromatin increases. Option 1.
Sure?
Heterochromatin increases, that I know, but differentiation of euchromatin increases or not, I don't know.
So heterochromatin increases, you are right. Whenever chromatin condenses during cell division, heterochromatin increases because heterochromatin is more tightly condensed form of chromatin, and whenever chromatin condenses significantly during cell division, the majority of the condensed material becomes heterochromatin, making it the primary form of chromatin that increases in this situation. And also in euchromatin is less condensed and it is generally where active genes are located. So here differentiation of euchromatin and heterochromatin decreases because it is saying that there is increase in the condensation of chromatin. No, I think there is some confusion here. It won't be option 3. Heterochromatin increases when there is an increase in condensation. Yes, when there is an increase in condensation of chromatin, the euchromatin, which is highly condensed, it highly condense and form the chromosomes. So distinction between heterochromatin and euchromatin is lost. So that's why the option 3, differentiation of euchromatin and heterochromatin, decreases. Okay. It is not heterochromatin decreases, it is saying that distinction between euchromatin and heterochromatin decreases. Because when there is condensation of chromatin, so it forms a chromosome. So distinction is lost there. Okay. Any doubts? Any doubts in this?
No, no.
Okay. The condensation of chromosomes is observed in
Prophase 1.
Anaphase.
condensation of chromosomes.
Second.
Anaphase.
Yes, ma'am.
Why? Condensation of chromosomes. How can it be in anaphase? Hmm? What happens in prophase? Prophase is the first stage of mitosis and where chromosomes condense. I hope you have heard of this thing. And at the end of prophase, nuclear membrane starts to develop, releasing the chromosomes. Although chromosome condensation fully completes in metaphase, but in anaphase, tell how can condensation of chromosomes occur in anaphase? It is prophase 1 where the condensation of chromosomes start and it gets completed in the metaphase 1. Anaphase 1 has no link with the condensation of chromosomes, okay? Clear?
Yes, ma'am.
In anaphase there is separation. It is not
Okay.
condensation. Moving to next, nuclear DNA replicates in which phase?
Option
G2MS.
3.
Option?
Option 3.
Option 3. Anyone else? Option 3?
Yes, ma'am.
Okay,
Yes, ma'am.
correct. Nuclear DNA replicates in the synthesis phase, right? Moving to next. Dash is a form of cell division which results in the creation of gametes or sex cells.
Meiosis.
Option?
2.
Option 2, right. There is spelling something else, so you have to choose the option. Okay, it is meiosis where the cell division happens and it results in the creation of gametes. Dash is the number of DNA in the chromosome at G2 stage of the cell cycle. 1, 2, 3 or 0.
Option 2.
Option 2.
Option 2. Sure? At G2
Yes.
stage, the number of DNA molecules is 2, right? Because during the synthesis phase, DNA replication occurs, which results in each chromosome having two identical sister chromatids, meaning there are two DNA molecules per chromosome in the G2 phase. Okay? So, option 2 is the correct statement. The stage which serves as a connecting link between meiosis 1 and meiosis 2.
Interkinesis.
Third.
Interkinesis. Everyone?
Yes, ma'am.
Yes. The stage that serves as a connecting link between meiosis 1 and 2, it is interkinesis, which is a short resting phase between two meiotic divisions where no DNA replication occurs. Okay. and it allows the cell to prepare for the second meiotic division by disassembling the spindle fibers from first division and reassembling the new ones. So in this phase is interkinesis. The longest stage in the cell cycle is longest stage in cell cycle.
First.
Interphase.
Yes, ma'am.
Anyone else?
Same only interface.
Yes. Interphase. It is the phase where cell grows and replicates its DNA, making it the longest phase of the cell cycle compared to the shorter mitotic phases where anaphase and metaphase are there. So, as it involves G1, S, and G2 phases where cell grows, replicates DNA, and prepares for the next division. So, it is the longest stage in the cell cycle. Which of the following state implies the exit of cells from the cell cycle?
G not.
Oh,
4. 4. Quiescent stage, right?
Yes, ma'am.
It represents a state where cells are not actively dividing and have exited the cell cycle. This phase is also known as the resting phase. Okay. It implies the exit of the cell from the cell cycle and it is also known as G node or quiescent stage. The cells exiting from the cell cycle enter the Z node stage after exiting the G1. After G1 it is G node. Now next question. Synapses is defined as the pairing of homologous, non-homologous, acentric, any chromosomes.
Homologous chromosomes.
Homologous. What is synapses?
Pairing of homologous chromosomes means the chromosomes which are somewhat similar, they will pair with each other.
Right. Homologous chromosomes are chromosomes that are similar in structure and have shared ancestry at a time. And during synapses, that chromosomes align alongside and allowing the genetic recombination to occur. This process which allows the matching up of chromosome pair, homologous pairs, before their segregation and possible chromosomal crossover between them. So this process is known as synapses. During meiosis, these chromosomes separate, which reduces the chromosome number to half. Okay. So synapsis is the pairing of homologous chromosomes which forms the bivalents. It occurs during which stage of meiosis? Remember?
Prophase 1,
And prophase 1, which stage?
zygote stage.
Yes, yes, very good. Mitosis can be observed in mitosis can be observed in which of the following individuals?
Ma'am, haploid individuals. But it can occur in anyone, not diploid also kind of mitosis.
Fourth.
Fourth,
Option 3.
only haploid.
Haploid, diploid, both.
And what about polyploid?
Also,
So option is it
option fourth.
fourth? What is mitosis? Here cell divides into two identical daughter cells with the same chromosome number as the parent cell, which can occur regardless of whether the parent cell is haploid or diploid. Okay, so if the parent cell is diploid, meaning they have two sets of chromosomes in their somatic cells, so after mitosis they will form diploid only, right? And haploid, if haploid which having one set of chromosomes, if mitosis occur there, then it will form haploid only, right? Mitosis can also occur in polyploid cells. It may be disrupted due to abnormal chromosome number, but it happens there as well. So all cells carry out mitosis. Only the sex cells, right? Not haploid individual I am talking about. Sex cells are the only cells which divide by meiosis. Okay. So option 4 is the correct answer here. Coming to next, the spindle apparatus is formed during which phase of mitosis?
Metaphase.
Metaphase. Sure?
Yes, ma'am.
Spindle apparatus. What is spindle apparatus?
Spindle fiber metaphase. Ma'am.
Ma'am, it will be in anaphase.
What is spindle apparatus?
Actually,
Spindle fiber attached with different poles.
ma'am, spindle fibers basically which are attached to the chromosomes.
So when this spindle apparatus is formed, formation of the spindle apparatus.
metaphase.
What is the correct answer? You have told everything: prophase, metaphase, anaphase, telophase.
First,
Telophase.
yes, ma'am.
Spindle apparatus is formed during which phase? It is asking about the formation, not the visualization.
Prophase.
Prophase. Sure?
Yes, ma'am. Yes,
Prophase is,
ma'am.
prophase is the correct answer, right? The formation of spindle starts during the late stage of prophase because the chromosomes start getting aligned during metaphase. So at the late prophase, spindle formation starts. That is why metaphase starts the spindle formation, and before metaphase, like when chromosomes have to align at that, before that, and in the late prophase, spindle apparatus is already formed. So its formation is completed during the earliest part of metaphase stage or in the late prophase. Actual late prophase formation starts. Okay, spindle apparatus needs to form with two of its poles so that later when the chromosomes can be arranged by it in the equatorial region. The starting of spindle formation only occurs in the case of late stage of prophase and are attached. Spindles are attached to the chromosomes, and in further stages they get separated. Okay, so here the correct answer is, it is showing metaphase, but actual correct answer is prophase. Spindle formation starts in the late prophase and it completes in the early metaphase. So if options are there related to late prophase or early metaphase, then you should choose. But here both are shown, so its formation is mainly in the prophase. You have to tick the prophase. Okay. Because in this phase the mitotic spindle begins to assemble as microtubules extend from the centrosomes, which start moving to opposite cell poles. So in metaphase they start to align there and it is shown, but formation of spindle apparatus happens in prophase. Moving on to next, cyclin is associated with which of these processes?
Mitosis.
Yes, if you were involved in the last class, you might have uh, heard about that cyclins and CDKs we learned about. So cyclins are the proteins that play a crucial role in regulating the cell cycle, and their primary function is to control the progression of mitosis by activating the CDKs, which is cyclin dependent kinases at specific stages of cell division. So cyclin is associated with mitosis. Coming to next, if an individual wants to view diakinesis, which of these would be? Anyone?
Onion root.
What is diakinesis?
Um, Diakinesis means it's in the final stage of prophase 1, where what means the chiasmata is formed. Uh,
Yes. Onion root.
yes, ma'am. Onion root.
And related to mitosis. If we want to see any of the mitotic phase, then which of these would you choose? Diakinesis is the phase in meiosis, right? And if you want to observe any of the mitotic phase, then which of these will you choose? Is it hair, leaf, onion root, or flower bud?
Mm, for meiosis, I think flower bud, and for mitosis, onion root.
Yes, yes. Now you are saying correct. For
Because
mitosis,
reproductive
yes,
organs are found in flower bud, so
yes, correct. So, diakinesis is a particular final stage of prophase 1 where chromosomes are fully condensed and visible, which makes it ideal for the observation. And flower bud is the best suited place to see diakinesis because meiosis happens in the reproductive parts of a flower. So, young flower bud is the perfect stage, perfect, um, perfect thing out of these to choose to view this diakinesis, because hair does not undergo meiosis and leaves also are not reproductive organs, therefore you would not see any meiosis here. Onion root is often used for the mitosis, and so it does not show the diakinesis, so the reproductive phase only diakinesis. Any of the meiosis stage, if you want to see, it would be floral bud, and any of the mitotic phase you want to see, it will be the onion root. Okay, so option 4. Coming to next, chromosome structure can be observed best during
structure means shape,
shape.
anaphase.
Sure.
Yes, ma'am.
Why?
Ma'am, because there the chromosomes move to the poles, so they form VLJI shaped.
Hmm. And what about metaphase?
Um, Metaphase, we can see the morphology, I think.
Chromosome condensation is complete.
Yes. In which phase?
Metaphase.
So best phase to visualize the structure is
Uh,
structure. What does structure of chromosome mean? it will involve shape, size, and number of chromosomes. So now tell which is the best.
I think metaphage.
Metaphase?
Yes, ma'am.
Okay. Correct answer is metaphase. Why? In prophase, what happens? Chromosomes are not clearly visible, right? Sister chromatids are intertwined with each other, and chromosomes overlap and appear like a ball of wool. Thus, ends of chromosome are not clear there. Now, coming to metaphase, chromosomes are condensed now. Hence, the centromere and arms of the chromatid are distinct and clearly visible. Due to this, metaphase is best stage to view any shape, size, or number of the chromosomes. Okay. Here, metaphase chromosomes align in the center of the cell, making them very visible under a microscope, and it is a useful stage for cytogenetics because chromosomes can be easily visualized. They are less condensed at early metaphase and becomes more condensed as the cell progresses towards the end of the metaphase. So the chromosomes carry genetic information and separated into each of the two daughter cells. So metaphase is a good time to count and study number and morphology of chromosomes. Okay. Now these are the assignment questions which you might have seen in your portal of NPTEL. So we will cover these 10 questions as well of week 4. How many 9 MERS and 13 MERS are in the Ori Sea of E. coli? Anyone? Any guesses? Heard about this E. coli origin of replication? No? 9 MERS and 13 MERS are there. How many 13 MERS and how many 9 MERS are there? So this is just memory based question. So it is, there are 9 13 MERS and there are 5 9 MERS. Wait, it is, yes, 5 9 MERS and 3 13 MERS, right?
Ma'am, ma'am, so option 1 and option 2 both are correct.
Yes, both are correct. So 9-mer sequences, what is the role of this? These are the binding sites of DNA A protein, which is crucial for initiating DNA replication. Okay, and 13-mer sequences, these are repeated sequences within 80 rich region of the RIC, which facilitates DNA unwinding during replication initiation. Okay, the origin of replication is known as RIC, and this region is where DNA replication begins. And there plays a role of DNA A protein, which binds to the 9-mer sequences and which initiates this replication process. So here 5 9-mer sequences and 3 13-mer sequences, which are located within 80 rich region. Okay,
Ma'am, what are these MERS, MERS?
these are like DNA binding on the DNA A binding sites. These are different proteins, like these 5 9-mers are R1, R2, R3, R4 and R5, and 3 13-mer binding sites including an 80 rich DNA unwinding element is there. Okay, 9-mers which show no differential specificity between DNA A ATP and DNA A ADP. That is quite higher part of this. Okay, we can go much in detail of this, but it will be according to your basic level. It is quite high because they then recruit something else, and it will go on in replication of this origin of replication of E. coli. It is quite high. Okay, so you just have to this remember this, that 3 13-mers and 5 9-mers are involved. Okay, and 5 9-mers will be R1, R2, R3, R4 and R5, and these 3 13-mers which will be included in 80 rich DNA unwinding element, which is also known as DUE. Okay, and 13-mers specifically recruit DNA ATP, and 9-mers show no differential specificity between DNA ATP and ADP. 13-mers will specifically recruit DNA ATP. So moving on to the next, the processivity of DNA polymerase in the polymerase 3 holozyme is increased by which of these subunit? This is also kind of memory based. Processivity of DNA polymerase. Anyone? It is beta subunit. Okay. So processivity of DNA polymerase in the polymerase 3 holoenzyme is increased by beta subunit. What beta subunit does, it forms a ring-like structure that tightly clamps around the DNA, allowing the polymerase to slide along the DNA strand without dissociating, thus significantly increasing its processivity. Okay, so it also acts as a processivity factor in the DNA polymerase 3 holoenzyme, and it forms a ring-like structure which encircles the DNA. It also enables the polymerase to synthesize long stretches of DNA without detaching from the template. So it is the beta subunit. RNA primers in the lagging strand are removed by which of these enzymes? RNA primers in the lagging strand are removed by which of these enzymes? Anyone? It is DNA polymerase 1. Okay. So what does it do? It removes the RNA primers from the lagging strand during DNA replication. This polymerase has 5 prime to 3 prime.
DNA polymerase one.
What? Are you saying something? What happened? So this particular polymerase 1 has 5 prime to 3 prime exonuclease activity, which means it can remove RNA bases. It also has polymerase activity, which means it can fill in the gaps between the DNA bases. And it is the most abundant DNA polymerase in eubacteria. It's also involved in the DNA repair by filling in the gaps between single stranded DNA. So how does it remove the RNA primers? It binds to the primer region first of all. As it moves forward, it degrades the RNA and replaces it with the DNA. And finally, the DNA ligase seals the nick in the phosphate backbone. You might have heard of the enzyme RNase H, which specifically identifies the RNA DNA heteroduplex, and it also removes the RNA bases. Okay. So here it is DNA polymerase 1. Centrioles are composed of how many triplets of microtubules?
Nine.
9. Sure?
Yes, ma'am.
This is also kind of memory based only. Centrioles are composed of 9 triplets of microtubules. Okay. It is a cylindrical structure with 9-fold symmetrical arrangement of microtubules. What is its function? What does it do? Centriole. It is involved in organizing the spindle fibers during cell division. Okay, you might have heard about microtubule triplets in case of basal bodies of cilia and flagella as well. Right? In which phase of cell cycle does centrosome duplication happen? Anyone?
S phase.
S phase. Sure?
ma'am.
Okay, it is S phase. So this S phase is when DNA replication happens. In this phase, DNA replication happens as it is a synthesis phase, and the centrosome, which is the microtubule organizing center, it also duplicates itself to prepare for cell division in the mitotic phase. So centrosome duplication happens in S phase. De novo centriole formation is associated with which of the following processes?
Cell division.
Cell division. Sure.
Means cell cycle. Cell cycle.
Sure. Centriole formation, and that too de novo. Anyone? It is flagella formation. De novo means from new, and it refers to the creation of centriole without a pre-existing template. Centrioles are essential components for the formation of cilia and flagella, as I already told. So when the new centrioles are formed, it is directly linked to the process of flagella formation. Centrioles act as basal body from which the flagella grow. So when centriole duplicate, you are right that cell cycle involves this, but is associated with which of the following processes? Centriole duplication occurs during the S phase of the cell cycle, that is true. But de novo centriole formation is associated with which process? It is different. Okay. That is centriole duplication happens. Now it is formed that from new the centriole formation is taking place. Okay, that happens in the flagella. Which of the following kinases induces both canonical and de novo centriole formation? It is memory based question, so without wasting time, it is polo like kinase 4. It is a kinase that induces both canonical and de novo centriole formation. It is a kind of serine threonine kinase that is a major player in centriole biogenesis. Okay, and it regulates both canonical and de novo centriole formation. And also, if I talk about the canonical pathway, this particular kinase triggers procentriole formation near a pre-existing centriole. Okay, while in the de novo pathway, it induces the cell formation when centrioles are absent because it is from new. In canonical, it is from the pre-existing centriole, and in the de novo, it is when the centrioles are absent. So overexpression of this particular kinase can induce de novo centriole formation, and depletion or inhibition of its kinase activity prevents the centriole formation. So any of the question can come that depletion or inhibition of polo like kinase 4 activity would lead to, then what will you take? It will prevent the centriole formation because this particular polo like kinase 4 is involved in the formation of centriole, both canonical and de novo. So it deregulation of this particular kinase is also linked to the tumor development as well, and it is a microtubule associated protein that self assembles. Okay, so moving on to next question. DNA replication in E. coli and other prokaryotes as well as eukaryotes occurs semi-conservatively, which implies which of these options?
Option 1.
Option 1. The strands of a DNA double helix separate and a new complementary strand of DNA is synthesized on each of the two parental template strands. Yes, you are right. What is DNA replication? And in E. coli and other prokaryotes as well eukaryotes, it occurs semi-conservatively. This implies that strands of DNA double helix separate and a new complementary strand of DNA synthesized on each of the two parental templates. So you might have seen the process that in DNA replication, like this, the strands are separated and new strands are formed. Like on one it is regular and on other it is Okazaki fragments which are finally sealed with the help of DNA ligase. And the strands of DNA double helix separate and a new complementary strand of DNA synthesized on each of the two parental template strand. Okay, so this DNA replication process occurs semi-conservatively. This implies this. Okay. Now, defect in which of the following proteins will lead to no replication fork formation? DNA replication. Few questions back we have covered the origin of replication.
Option 2.
No replication fork. It is not asking that no replication, it is asking no replication fork. So, which of the following proteins or enzymes would be involved in the formation of replication fork first of all, and defect in which of the following will lead to no replication fork? What is the function of DNA polymerase 1? It has this activity 5' to 3' exonuclease which we heard about, and it removes the RNA primers, right? Now 3' to 5' exonuclease activity also. So in replication fork formation, which of the following enzymes is there? it will be DNA A protein. Okay. In few questions back, we have taught learnt about that DNA A protein is responsible in RIC, where I was talking about 9 mers and 13 mers. So I told that in RIC it is responsible. So this particular protein is crucial for initiating DNA replication by binding to the RIC, and it is essential for the formation of replication fork. Okay. And DNA polymerase 1, this is crucial for DNA replication, but its primary function is to remove RNA primers and fill in gaps not directly involved in the initial formation of the replication fork. It is involved in the replication but not in the replication fork formation. And also the 3' 5' exonuclease activity, it is for the proofreading purpose only, which checks for the errors during replication. It is not directly linked to the fork formation. It is only the DNA A protein which will lead to the, which binds to the origin of replication, that is RIC, and it is responsible for the formation of replication fork. Okay. And if there is defect in this protein, there will be no replication fork formation. Okay. So without a functional DNA A protein, the replication process cannot even begin. If we have DNA polymerase and we have this exonuclease activity of another DNA polymerase, still the DNA replication will not begin if there is no formation of replication fork. First of all, DNA A protein goes and binds to the RIC, then only replication fork begins and DNA polymerase starts its function. Okay, so remember this thing. This is very important question. Moving on to the next, 1000 base pair long double-stranded DNA molecule contains 30% adenine. Determine the number of guanine bases. This is easy. थाउजेंड बेस पेयर में 30% एडेनिन. हाँ जी.
Option 3, 200.
Option 3. So 30% of 1000 will be
300.
300. So 300 will be your adenine A. So T will be also 300, right? So left over 300 and 300, total 600 will be 80. Now GC will be 400, right? 400 will be your GC. So each will be, now it is asking about guanine bases, so it will be 200. 200 of guanine, 200 of cytosine, 300 of thymine. Any of these will be asked, any percent could be asked. So this is easy. So talking about the basics of mitosis and meiosis before after ending the questions here. Mitosis, it is a when the cell wants to duplicate itself. If any of the cell wants to duplicate, to make a duplicate of itself, first it must copy its DNA, right, as a part of a chromosome. The copies then must be separated and sorted into two sides of the cell, right? In the like cell, when cell wants to duplicate itself, it must copy its DNA. So the copies must be separated to two sides of the cell. The cell then splits into two. Part of each parent is carried into two new cells. Results in cells such as internal organs, skin, bones or blood. Now talking about meiosis, as in mitosis, if a cell wants to make a duplicate of itself, it must first copy its DNA. Results in reproductive cells, here sperms or eggs. Okay, so part of each parent is carried to the four new cells. Here each will be in the 4. Meiosis has evolved to solve a problem. The problem is this: some organisms mate with other organisms, that is, they combine their genes together, creating genetic diversity. Now, why would this be a problem? If the two organisms mate, so the cell number, chromosome number would be duplicated. If meiosis won't be there, then it would go on increasing. So mitosis is the makes it 2. Meiosis is has role in the sexual reproduction. Okay, so mitosis, it means identical twins, and meiosis means egg and sperm, which leads to formation of haploid gametes. Okay, so mitosis, when there is this kind of like male and female gametes, each progeny will carry these copies. And when in meiosis, first of all they will separate in meiosis 1, and then meiosis 2 will lead to formation of 4 haploid gametes. Each resulting cell still has chromosomes from mom and dad both. So starting from the mitosis, there is interphase which occurs just before the mitosis begins. Here DNA is replicated along with organelles and other cellular components, and the cell prepares for division. Here centromere is there, where replicated chromosome, and here 2 sister chromatids are there. Here in the interphase, in the mitosis, this is how animal cell looks like, and here plant cell. And in the first stage of mitosis, in the prophase, which is the preparation phase, here everything starts to disintegrate. The DNA recoils, chromosomes condense, nuclear membrane disappears, and mitotic spindle begins to form. This question we have already covered, that mitotic spindle begins to form in prophase. Mitosis. Now metaphase. The chromosomes line up in the middle of the cell with the help of spindle fibers. This question also covered that spindle fibers formation, spindle apparatus formation begins in the prophase, and finally in the metaphase the chromosome lines up in the middle of the cell with the help of these spindle fibers, which attach to the centromere of each replicated chromosome. This is how it looks like. Now in the anaphase, there is separation of these chromosomes which split in the middle, and sister chromatids are pulled by the spindle fibers to opposite poles of the cell. In animal cell and plant cell, how it looks like. And in the telophase, finally, the chromosomes along with the cytoplasm and its organelle and membranes are divided into 2 portions. This diagram shows the end of the telophase, where after this what happens? Next phase is after telophase. After telophase, what will happen to separate these? Telophase, this is how it looks like, and after this cytokinesis happens, and in the plant and animal, in the plant it begins from the center, and in the animal from the outside. How this furrow begins? In the animal it happens like this, and in plant like this. The actual splitting of the daughter cells into 2 separate cells, it is known as cytokinesis, and it occurs differently in both plants and animals. Now Now meiosis, meiotic division 1 and 2 in first and second. So starting from the interphase, the chromosomes have yet not condensed. Here the chromosomes have replicated and chromatin begins to condense. These are finally completely condensed. In meiosis, the homologous chromosomes pair with one another. The nuclear membrane dissolves in the metaphase and homologous chromosomes attach to the spindle fibers. They are preparing to go into opposite poles in the anaphase. So in the metaphase they are on this plate, metaphase plate. And anaphase, finally the chromosomes move to opposite ends of the cells. Now cytokinesis happens and it divides to form two daughter cells having both maternal and paternal chromosomes. Now coming to meiosis 2, that is prophase 2, the cell has divided into two daughter cells. Metaphase 2, as seen in meiosis 1, the chromosome line up on the spindle fibers. The two cells each begin to divide, as in meiosis 1, the chromosomes move to opposite ends of each cell. So within this formation of four cells, meiosis is over and each of these prospective germ cells carries half the number of chromosomes of somatic cells. So here ending this mitosis and meiosis. I hope it is clear. Any doubts still here? So today we have covered the extra questions as well as assignment questions of week 4 and also covered the mitosis and meiosis. So for any queries you can ask next time as well. So today I am ending here. Thank you for joining.